welcome back to this channel where hopefully I have calmed down, because last video I was way too nervous and stuttering way too much, and hopefully that is all calm by now and we won't have any problem. Now last video I forgot to tell you guys how you are supposed to prepare and watch my videos. So all you need to do is get your textbook and a note. If you're not on your laptop, just get a paper notes, and whenever I'm on the full screen like right now, hey, you give all your attention to the screen, to me right now. And whenever I am referring to a diagram, you just pull up that diagram or that topic inside your textbook and you go through that and you start taking notes. So what are we doing today? In this chapter we are going to be discussing the first week of development after the fertilization, how an zygote just goes from day 1 to day 6 throughout the first week of development and all the processes that comes through it. Of course I'm gonna be in depth and I'll try to make it as easy as possible for anyone to learn. So without further ado, let's jump right into it. As you can see this diagram, this is the only diagram we need for this chapter. Week 1, this diagram contains each and everything you need to know, and I will go through it bit by bit, and in the end we will get to day 6 where implantation happens all the way from day 1 or day 0. So as you can see here, here's the ovary where all the eggs are. Now each and every month one egg is released from the ovary to the ampulla. Ampulla of the oviduct. Ampulla is also the opening or the end of the oviduct. That's where the egg is. Now one important thing to notice here is the egg does not get released directly into the ampulla. Now from here there's a gap, of course, everything peritoneum, the abdominal cavity where the egg is released, and from there the charge distance, and with the help of small villi the egg is sucked into the ampulla of oviduct. Now let's talk about some complication. Ovary releases an egg in the peritoneum cavity, but let's just say for some reason it is not sucked into the ampulla of oviduct. Now what happens? The sperm just travels from all the ovarian tube to the ampulla of oviduct where it is supposed to fertilize the egg, but the egg isn't in the ampulla of the oviduct. So what happens? The sperm just doesn't stop there. The sperm keeps swimming and it will soon reach the peritoneal cavity too. So both the egg and sperm, they are in that cavity, and of course they can fertilize there. So that is a kind of abnormal pregnancy, ectopic abdominal pregnancy. Now not to be confused with the other kind of ectopic tubular pregnancy. Ectopic, what does that even mean? Ectopic just means something that is out of its place. So it was supposed to be in the ampulla of oviduct, but now it is in the peritoneal cavity. So ectopic abdominal pregnancy, simple as that. Ectopic meaning it's out of its place, abdominal where the pregnancy is, simple as that. Now that we have covered that, let's say if thing goes by normal ranges and the egg actually makes its way to the ampulla of oviduct. So what would happen? The sperms will swim all the way from the tube to the ampulla of oviduct where it is supposed to meet with the egg and it is supposed to fertilize it. Now how does that happen? We need to focus on this part of the diagram for that. Secondary oocyte arrested in metaphase of meiosis 2. Now we already discussed meiosis 1 and meiosis 2 in the previous chapter. As I explained in the chapter before this, meiosis 2 in female only occurs when an egg is fertilized by the sperm. So when fertilization occurs, that is when the stage meiosis 2 happens. So as you can read here, secondary oocyte is arrested in the metaphase of meiosis 2, and when the sperm actually makes its way through it, that is when the meiosis 2 will start and end in a female, and after that we are back to the above mitosis like I explained in the chapter above. So how does that happen? Let's talk about that. As you can see here, the covering layer of secondary oocyte is zona pellucida. As you can notice here, the zona pellucida is supposed to be the protection layer of secondary oocyte. Now how does the sperm infiltrate it? The head of the sperm, which is also known as the acrosome, the head of the sperm will release enzymes which will create a reaction and dissolve this particular layer, the zona pellucida. Now once the penetration is done, it reacts in a cortical reaction which prevents the other sperm from entering and fertilizing the same egg. Now once the process of fertilization is done, the sperm infiltrates the zona pellucida and finally fertilizes the egg. That is when the fertilization occurs. It is supposed to be in the ampulla of the oviduct. That's where it's supposed to happen normally. So once that is happened, the process of meiosis 2 will start. Now as I said in the previous chapter, after the process of meiosis ends, where does it go? It goes back to level 1, where it divides by the process of mitosis, not the meiosis, mitosis. So that's where we are. Day 1, that's where we are. This one cell is called zygote. This zygote, as I mentioned in the previous chapter, before process of meiosis 1, this zygote will divide by the process of mitosis. Now in any other organ, let's just say for liver, intestine, stomach, any other organ, skin, the process of cell division through mitosis happens like this. One cell divides into two cells, they both grow up and then they divide into two other cells. So from one cell you have two cells, they both grow up, then they divide equally, and then you have four. From 1 to 2 to 4, it just goes on to multiply. But in this, the cell divides by the process of mitosis. 1 goes on to become 2, 2 goes on to become 4, 4 goes on to become 8, and so on. But in this particular case, the cell do not go through the process of growing up before. So they do not enlarge and they do not develop, they do not grow before dividing again. So as you can notice here, the one cell goes on to become two cell goes on to become 4, 4 goes on to become 8, 16, 32, 64, and so on. But as you can see here, the size of all the cells, the zygote is a big one. The two cell blastula is divided into half, they do not develop further, so it's half the size the zygote was, and the same goes with day 3 4 cell blastula. So it is dividing but it does not develop, it does not enlarge, and that process of mitosis, it's what we call cleavage mitosis. It only occurs here. In any other normal tissue or cell, the process of mitosis will just go through as planned. Now another interesting fact, once we are at day 4, we either have 32 or 64 cell. That day 4 is what we call as a morula. It doesn't matter much if it's a 32 or 64 cell, but on day 4, that stage is what we call as morula. After we have morula and the cell keeps dividing, another process occurs before or on day 5. All the cells, they just come together, regroup, and then they just push each other, or they just are in the side, and the majority of that cell cavity is filled by fluid. Now when that process happens, that particular cell on the day 5, we would refer it as a blastocyst. As you can see here, day 5 it's a blastocyst. Day 4 it was called a morula. It hadn't begun the process of just dividing into just pushing all the fluids on one side and having all the cells on the other side. But when that happens on day 5, that is when we would refer it as a blastocyst. Now let me make it even more easier for you. When you hear the word blast, not the actual blast, when you hear the word blast in embryology, that would refer to as a cell which will go on to become different type of cell. So as you can see here, embryoblast, so that just means cells, blast cell which would go on to become the embryo. So it is self-explanatory. Embryoblast forms embryo and tropoblast forms placenta. So the blast would go on to become different kind of cells. So whenever you hear the word blast in embryology or anatomy, you would know the cell will go on to become a different kind of cell when it's developed. Now on to day 6, when the process of implantation is supposed to occur. So going back to day 1, remember when I said when we can have an ectopic abdominal pregnancy, when the egg just releases into the peritoneum cavity, then it will still fertilize, and on day 6 the egg or the zygote, or we call it blastocyst or whatever, that will of course attach itself to another surface. It doesn't matter, it doesn't have to be the wall of uterus, it will have many different surfaces. It will attach to a surface regardless, and when it actually develops the process, the walls will not have the smooth lining which helps pushing out this when giving birth. So if you have an abdominal ectopic pregnancy, that will result in complication because of the bleeding and you don't have the smooth muscles to push it out, and that is why usually the ectopic abdominal pregnancy is aborted. During day 6, the process of implantation begins on the wall of uterus. It will usually attach itself in the posterior wall of the uterus. But let's talk about another complication. On day 6, let's say the cell and the egg has fertilized, it goes on to develop here day 2, day 3. But let's say if the cell has not made to the wall of uterus, what would happen then? Let's just say the cell is stuck here or whatever here. Then what would happen after? That would usually occur when we have a scarred wall lining. Scarring of the wall of the tube usually hinders the movement of the cell into the wall which is supposed to be on day 6. So that kind of pregnancy is also ectopic because it is out of the place. It's not where it's supposed to be, so it would still be ectopic, but instead of the abdominal, it is now called the ectopic tubular pregnancy. Now, how does the process of implantation occurs? We will go deeply into that. As you can see here, the embryoblast and the tropoblast. The embryoblast will develop into divided more cells, but the tropoblast will differentiate into two different kinds of cell. As you can see here, the cytotropoblast and the syntheotropoblast. What is the difference, you may ask? Cytotropoblast forms the lining of this blastula, but the difference between cytotrophoblast and syntheotropoblast is syntheotropoblast will have finger-like projection, finger-like villi, which will erode the wall of uterus, and that is the process of plantation. Now, syntheotropoblast, those villi-like structure, they do not divide. Unlike cytotropoblast will divide, but some of the cytotropoblast will fuse together and which will form the syntheotropoblast. Syntheotropoblast do not replicate or reproduce or go through the process of mitosis. Only cytotropoblast, they go through, they divide, they multiply, and cytotropoblast, when they are fused together, then syntheotropoblast is formed, and they will adjust and they will hook themselves to the wall of the uterus. They will just erode their way through and implant into the wall of the uterus. Now, another important thing, let me get my note up for this. Now, as you can see here, the syntheotropoblast synthesizes an ACTH-like peptide which is called the human chorionic corticotropin, HCC in short. You might have heard it before. HCC is usually referred to when we are detecting a pregnancy. You might have heard the term before. So HCC, the human chorionic corticotropin, which will tell the mother to retain the corpus luteum. Now, what is corpus luteum? Let me just divide it for you. Corpus luteum. Corpus means body and luteum is yellow. So it is a yellow tissue which is released every month after the egg, and it just degrades after few days and then it is flushed away. Now, degrading only stops only and only if the egg is fertilized, otherwise it will just degrade away. So we need to maintain the corpus luteum in order to maintain this pregnancy. So corpus luteum, which is master cell that is responsible for the production of hormone progesterone during early pregnancy. Now, this progesterone is the one that is going to maintain the pregnancy and maintain the endometrium. And that concludes the week 1. We go through, let me just revise it quickly for you. The egg goes through from ovary into the peritoneal cavity. From there it is sucked into the ampulla of oviduct. After that it is fertilized there and goes through the tube. On day 1 it is a zygote, day 2 it's a 2-cell blastula, then 4, and 8 and 16 until it is either 32 or 64, and then it is called a morula. After that, all the cell just pushes into a single direction and the other rest is filled with fluid. The cells are called the embryoblast and its covering layer is called the trophoblast. Trophoblast divides into cytotrophoblast and syntheotrophoblast. Cytotrophoblast divides, and when they fuse together, that is when they form the syntheotrophoblast. After that, day 6 is when the implantation begins. Syntheotrophoblast erodes into the wall of uterus and just sticks themselves there, and then they produces HCC, which maintains corpus luteum, and then corpus luteum produces progesterone, and that maintains the pregnancy. That concludes week 1 of the development. I hope that was helpful to you guys. Hopefully I explained in depth, and hopefully it was helpful for you guys. Tell me if I can improve, tell me how I can do it, and if you have any questions, just write them in the comment below and I will reply to you guys. Hopefully you guys enjoyed this video. I will see you when the next chapter drop. Goodbye.